So this is our last week of lectures. Next week is Exam 5 on Monday at noon, Tuesday at 4 o'clock, and you guys are invited to attend either one. And then the week after that is Exam 6 during finals week, and it's also Monday at noon or Tuesday at 4. So again, you're invited to attend either one. Okay. Chapter 29 in our text is titled Development, Pregnancy, and Heredity. And let's go ahead and talk about some terminology here. And that's what the definitions are all about. Oocyte, female gamete, commonly referred to as egg. Spermatozoon, that's singular. Spermatozoa is plural. It's the male gamete, commonly known as the sperm or sperm cell. Pregnancy is referring to prenatal time period, and this is a maternal event. When the word pregnancy or prenatal, we're talking about what's going on with mom. And pregnancy is from fertilization to birth. Then gestation is what the term that we are using when we refer to the time of development of the conceptus. And conceptus is a very generic term. Very generic term like thyroid medication. There are various kinds of thyroid medications and name brands and various generics, but gestation conceptus is a very generic term. And we can use it to describe the zygote that we see in the preembryonic period, or the embryo in the embryonic period, or the fetus during the fetal period. So very generic term, but referring to the products of conception. So gestation is a terminology that we'll associate with that. Preembryonic period, first 2 weeks after fertilization, we can still call it a conceptus, or we can use the term zygote. So zygote is much more specific. So when I say zygote, you're going already, that's the preembryonic time period from fertilization through the first 2 weeks. The embryonic time period is going to be weeks 3 through 8 of the developing conceptus, and the fetal period is going to be weeks 9 through birth. And so you've made certain advancements in your development whenever you go into these various periods. Let's take a look at the first gestational period of the conceptus, the preembryonic period. We're going to go from egg at the beginning, and then when we terminate this preembryonic period, we are known as an embryo because we're entering weeks 3 through 8. This is a really nice scanning electron micrograph of an oocyte and some spermatozoa, and it shows you the relative size of the two. So the oocyte is huge, full of cytoplasm for the first 2 weeks of this preembryonic time period, because we aren't going to be reliably retained until we've undergone successful implantation. So that will allow us to go to the next stage, our embryonic period. Nice big egg or oocyte and small sperm. The head of the sperm is where the DNA is actually been condensed and stored for this transport process. We're going to go into more detail about this process than what we have with our cartoons here with phase 1, phase 2, and phase 3, but let me just kind of preface what we're going to go into with phase 1. Phase 1 here, sperm penetrates corona radiata. So it's referring to this area close to the end of my arrow there. What do you also know the corona radiata to be called? What is a general term for where it came from? What cell line? They used to be granulosa cells. So they're still granulosa cells, but when I say corona radiata, you know that those are the granulosa cells that are immediately around the oocyte. So sperm penetrates corona radiata. These are cells, as you can tell, they're cuboidal cells producing estrogen because they're still in contact with the oocyte, and they have cell-to-cell connections. And so the sperm cell has to break through those cell junctions in order to actually get into the next area, which is going to be our zona pellucida. So zona pellucida is internal to the corona radiata and is a very important layer that the sperm cells have to interact with in order to actually break through this. And so we see here in phase 2, it says sperm undergoes acrosome reaction and penetrates zona pellucida. So the sperm cell, as we go through our notes, we're going to learn when it comes into contact with the zona pellucida actually has sperm cell receptors, and the sperm cell contacts that sperm cell receptor of the zona pellucida. A reaction is going to occur that will allow the acrosomal enzymes at the front of the head of the sperm to suddenly start rupturing out and pounding or chemically digesting a tunnel through the zona pellucida. So the zona pellucida has a length or depth to it. And so the sperm cell has to penetrate that in order to get to the oocyte. And that's phase 3. Sperm and oocyte plasma membranes will fuse. When that happens, it also stimulates the completion of meiosis 2, and then we end up with another polar body. So here we have in this 2 polar bodies, and whenever you see 2 polar bodies, you should realize meiosis 2 has completed. We have this 1 polar body here that was a product of completion of meiosis 1. And the polar bodies are meant to not be fertilized because the thing that's meant to be fertilized is the actual oocyte or egg. And then we see here as part of the notation, sperm pronucleus and ovum pronucleus, and we'll talk about these pronuclei also. So this is just a little intro into what we're going to discuss as we go into the notes further. Fertilization. It's important to realize in order to fertilize an egg, your oocytes are only viable for 12 to 24 hours after ovulation. The other important thing to realize is that the sperm cell, once deposited into the female reproductive tract, is only viable for 48 to 72 hours. And our zygote, which is our newly fertilized egg, or again, the generic term conceptus, is what we want to actually create with fertilization. But in order to do this, in order to have successful fertilization, knowing the times of viability of the oocyte and sperm are important because it tells us the time frame for proper semen deposition into the reproductive system of the woman. So semen deposition must occur no more than 3 days before ovulation and no later than 24 hours after ovulation. This is actually known by a very certain religious group that has a method of practicing birth control based upon ovulation time of the woman. And this is very common for people who are Catholics to practice what is called the rhythm method. And so they know, the woman knows when she ovulates that if they want to have a child, then they should have intercourse as soon as they can after ovulation. She also knows that if she doesn't want a child after ovulation, she doesn't want to have intercourse before ovulation during that time period, the 24 hours. And also, if they happen to have intercourse within a day or 2 before she ovulates, she knows that she is susceptible to becoming pregnant if she doesn't want to actually achieve a pregnancy. And so what they typically do when people practice the rhythm method, they actually abstain or use condoms for intercourse during that 3 days before ovulation or 24 hours or a day after ovulation. So, and it is fairly successful, but not like the birth control pill. the success rate of the birth control pill is far superior. So now we understand the viability of the egg and the sperm cell. And now let's go ahead and talk about this process of sperm transport through the female reproductive system and how it actually is important to actually create a sperm or sperm cells that are capable of fertilization. And capable of fertilization must have been where they came up with the term capacitation. Realize, and we're going to use an average ejaculate. In an average ejaculate, there's about 350 million sperm cells. It's interesting that only a few 100 actually make it to the egg for fertilization. So this is telling you that there is a high attrition rate. And we're going to actually go through some of the reasons for this high attrition rate or loss. Sperm cells are deposited in the female vagina, and in that vagina, not all of the ejaculate will remain. After deposition, and we know that the pH of the vaginal area is quite acidic. There's still a lot of spermatozoa that are actually able to make it to the cervix. But there's something about the cervix that is another barrier that they have to actually be able to traverse. Do you guys remember what protects the woman from things of the outside world from getting inside to the uterus spot, uterine body? Mucus. So there is a very thick mucus plug in the cervical canal. And so we have that mucus plug thins with high estrogen levels. Well, when are the estrogen levels high? During ovulation, right? So ovulation is the high estrogen levels is the stimulus for ovulation, and high estrogen levels also thin out the cervical mucus, and it becomes quite watery. So the sperm cells are able to traverse that much easier than being able to penetrate a very thick mucus plug. So what's nice is that it goes through the cervical canal, but the cervical canal has lots of nooks and crannies, and actually several of them get lost in the nooks and crannies of that cervical canal. So we can say areas in the canal traps spermatozoa. So they go through the external os, through the canal, and then through the internal os, and now they have made it into the endometrial lining area or lumen of the uterus. And in the uterus, the sperm cells are treated as foreign bodies. And what do we have that's part of our innate immune system that doesn't like foreign bodies and gets rid of them for us? Macrophages. And so we have lots of macrophages inside the uterine lumen, or adjacent, obviously, to the mucosa. So we lost some in the vaginal area. We lost some in the cervical canal. Now they're inside the lumen of the uterus, moving along the mucosa of the uterus, being taken out by various macrophages. And then they also have another thing that will cause a diminished number of these cells making it to the oocyte or egg. And that's because there are how many uterine tubes? 2. And so some go in the right, some go in the left. And so they'll go down the wrong uterine tube. So that further affects the attrition rate, or the loss of the sperm cells. Now, once it gets into the uterine tube, what do you know about factors that are occurring in the uterine tube that work against the sperm cells also? Peristalsis and cilia. And so are they going in the direction, helping the sperm go towards the oocyte, or are they swimming upstream, so to speak? They're swimming upstream, so they're swimming against a current. So that also diminishes their ability to make it to the oocyte. And also there's a lot of villi in the fallopian tubes too, for them to get actually lost in also. So one of the things that's interesting though, the sperm cells that make it to the oocyte have undergone a tumultuous process. They have been roughed up, wouldn't you say, on their way to this? And the roughing up is exceedingly important for this process called capacitation. So capacitation, our notes say it takes several hours, but what this process involves is the sperm cells undergo physical and physiological changes which allow them to actually be able to become able to actually fertilize an egg. It's kind of like their boot camp. So they're in this process and they're undergoing all kinds of assaults, so to speak, and it toughens them up, but not really. It actually causes their acrosomal enzymes to actually be more prepared or ready to actually be released when they get to the zona pellucida. The important thing about this process is if we take sperm cells out of the epididymis of a mature male, remember sperm cells are there for maturation and storage. And so maturation is a normal sperm appearance and sperm motility and all that kind of stuff involved with that. But if you take those sperm cells out and then put it on a petri dish with an oocyte ready for fertilization, they will not be able to fertilize that egg. So they have to undergo all of this interference with their forward movement to actually cause them to create their ability to actually have what's going to be called the acrosomal reaction. And that's what capacitation is all about. It's the traveling through the female reproductive system that actually makes them capable of actual penetration fertilization of the egg. We're going to take a look too at some other barriers that the sperm cell is going to have to be able to break through to encounter the egg. And we have 3 layers here. We have the cumulus, the corona radiata, and the zona pellucida. So say we have about 100 sperm cells that have made it to the ampulla of the uterine tube. The ampulla, remember, is a swollen area where fertilization is supposed to actually occur. That's the optimal site for fertilization. With ovulation, one of the things that actually you have to realize, before ovulation occurred, what did we call that follicle? It's called a mature or vesicular follicle. And it has that corona radiata around it in the follicle, but it also has this fluid around it. You guys remember that we actually have fluid that is starting to fill up in areas of the granulosa cells. With the mature or vesicular follicle, we have a lot of fluid, and that fluid actually still goes with the oocyte when it is actually ejected or ovulated from the ovary. And that fluid is called the cumulus. And so here it says the cumulus is a cloud-like layer of sticky material. What's really nice about this is that it is chemotactic for sperm. So in addition to actually being able to penetrate this bit of sticky material or viscous material, this material in itself is actually telling the sperm cells, go down this fallopian tube or uterine tube. Those that are able to actually respond to it appropriately. So cumulus helping us to get there, but also still a slight barrier. The next barrier still that must be penetrated is the corona radiata. And remember, those are granulosa cells that are encapsulating the egg, and the sperm must actually create a pathway through the corona radiata so that it can get to the next layer, the zona pellucida. So they're having to wiggle through cell junctions to actually break them down and to actually get on into the next layer. And what's really interesting, they actually have an enzyme called hyaluronidase, which is actually designed to actually break through cell layers. This is also a component in a lot of women's moisturizing makeups for their face, things along that line. The zona pellucida, they've made it through the corona radiata, created a path through that, and now we've got this tough glycoprotein coat, sugar protein coat, that's directly over the oocyte membrane that also needs to be breached. So with this zona pellucida, what's important is first sperm cell to arrive and undergo acrosomal reaction just makes a dent in the zona pellucida. So we have about 100 players there. They've made it through the cumulus and the corona radiata. And the first one that gets to the zona pellucida will have an acrosomal reaction. Boom! We'll have some acrosomal enzymes released and we'll make a dent in the zona pellucida. What does that mean for that sperm's ability to actually fertilize that egg? He's going to go by the wayside. So then another sperm cell is going to go into the very same channel, so to speak, and boom, have another acrosomal reaction, and again, create a bigger dent in the zona pellucida. But that one also is not going to be able to actually fertilize the egg. And so you actually have to have several acrosomal reactions, therefore from several sperm cells, that actually break through the zona pellucida to actually get to the egg for fertilization. So first sperm cell to arrive doesn't fertilize the egg, therefore does not fertilize. We actually have to have many acrosomal reactions to do this. So let's go ahead and take a look at what the acrosomal reaction is. It occurs at the zona pellucida because the zona pellucida is kind of interesting in that it has sperm cell receptors. When the sperm cell comes into contact with the sperm cell receptors, sperm calcium levels actually rise. So this is due to spermatozoa coming into contact with ZP sperm cell receptors. The rise in calcium levels will then trigger the acrosomal reaction. Uh-oh. That's a bad sign. Oh dear, this is not going to go well. Acrosomal enzymes from the acrosomal cap, which is in front of the DNA that the sperm cell is carrying, and they're released and begin digesting a pathway through the zona pellucida. So we finally have a sperm cell that has now a clear pathway to the oocyte membrane. And so that will allow us to have what's going to now be sperm penetration. And that did require many acrosomal reactions for this to occur. And the first sperm cell to make actual contact with the oocyte membrane will have its membrane fused with the oocyte membrane. And that's just going to be a small spot on that oocyte, because remember how large that oocyte was, and then the sperm cells actually of the head is actually very, very small. Now, once this sperm penetration actually occurs, we're going to have some reactions follow to prevent this process here called polyspermy. So the DNA packet of the sperm cell is now delivered into the interior of the oocyte because of the fusion of the two membranes. And we only want to have one packet of DNA from sperm cell to actually go in, correct? Because that packet has 23 chromosomes and the oocyte has 23 chromosomes. If we had two sperm cells actually deliver their packets of DNA to the oocyte, that means we would have 3 sets of 23. And does that allow for any viability of this conceptus? It is not viable. We can have maybe an extra chromosome here or there, but we cannot have a whole set of extra chromosomes. We are designed to be diploid, not triploid or tetraploid. We are designed to be diploid as human beings. And so we want to then stop any other sperm cells from doing this. And that's what blocks to polyspermy is actually all about. We want to keep our ovum viable because ovum is rendered non-viable if more than one sperm cell penetrates and delivers its packet of DNA. So we want to actually keep this conceptus, soon to be conceptus, viable. And so this is what actually happens. It's called zona reaction, and this is because it happens to the zona pellucida. That sperm nucleus entry is very important because it causes the oocytes endoplasmic reticulum to release its calcium. So this is a calcium-driven process for acrosomal reaction for the sperm cell and a calcium-driven process to block polyspermy at the site of the with the oocyte. This rise in calcium, which is being released by the endoplasmic reticulum, is going to cause this next step. It's called a cortical, remember cortex is the outer aspect, a cortical reaction. And what happens is on the inside of the oocyte, there's a whole bunch of enzymes hanging out right on the inside of the oocyte membrane. And when we have this calcium release, these enzymes are then actually forced or put out into the extracellular space. They're actually put out between the cell membrane and the zona pellucida. These enzymes have an effect on the zona pellucida like air does on Fix-A-Flat. Do you guys know what Fix-A-Flat is? It's that stuff that's in a can, and if you get a flat tire, you put it on into the little, I want to call it pin. What do you call that thing? Valve stem. Thank you. Valve stem, and you push it on in, and suddenly it actually fills it up and it expands and creates a temporary firm surface so that you can drive to, a gas station or a place to actually get your tire replaced or repaired. So Fix-A-Flat's really interesting. You expose it to air and suddenly it expands and firms up or hardens. This is what the zona pellucida does. So the zona pellucida, because of the calcium that is being released by the endoplasmic reticulum, forcing these enzyme granules into the extracellular space, these enzymes interact with the zona pellucida and cause it to swell up like Fix-A-Flat and harden. So if you are a sperm cell making it through the zona pellucida and this happens, what happens to you? You're trapped inside this, what I call Fix-A-Flat, and you go nowhere. What about the sperm cell receptors that were associated with the zona pellucida? Do you suppose that they stay the same? This actually destroys also the zona pellucida sperm cell receptors too. And that is what the cortical reaction is all about. Enzymes are released, they interact with the zona pellucida, cause it to swell and harden, and also destroy the sperm cell receptors associated with the zona pellucida. So I like to, and if you don't know what Fix-A-Flat is, you probably should get a can for your car. It's nice to have on hand. Zona pellucida sperm cell receptors also mean no more acrosome reactions. So that's how we block polyspermy. First one enters, calcium levels rise, enzyme granules go to the extracellular space of the oocyte, come into contact with the ZP, and swells up, and now we got a nice protective shell around what is becoming now our fertilized egg. Because in order for fertilization to complete, we actually have to do this first. We have to have completion of meiosis II in order to actually fertilize. We can't be stuck in meiosis II. And so what's important is this calcium release also triggers the completion of meiosis II. So once meiosis II is completed, the rest of the fertilization process will ensue. So completion means that we have one polar body and a nice big fat oocyte, correct? That's what meiosis I completion gave us, right? Nice big fat oocyte and a polar body. So then completion of meiosis II is going to cause that oocyte to finish meiosis II, give us a nice big fat oocyte and another polar body. Now, this is all stuck inside this shell. The zona pellucida now has become a confining shell. What's important is we got to get the nuclear material together in order to actually start mitosis. Because mitosis is, now we have two sets of chromosomes and we're going to undergo cell division with mitosis. So they call the material that's inside the oocyte from the male and also even the female, but more importantly the male DNA is very tightly compacted. It's so tightly compacted we have to unravel it and actually allow it to swell so that it can actually then get together with the female pronucleus and then go to the mitotic spindle for replication. So yeah, that's why it says male and female pronuclei swell. It's really very important for the male pronuclei to swell because it's really super small and compact. Mitotic spindle will form now, bringing the pronuclei together. And once we bring the pronuclei together, we can then start replication. So we have one set of chromosomes for mom, one set of chromosomes for dad. They swell up to where they're recognizable. They are actually brought together. And once they're brought together, they undergo that replication process. And so mom's chromosomes replicate, dad's chromosomes replicate. And you guys remember the two arms of replication? They're called chromatids. You guys remember that? And then we have the centromere that holds them together. So now we have all of these chromosomes from mom and dad replicating, going to the equatorial plate, and then we undergo then the separation to anaphase and telophase. You guys remember that? And then we end up with the formation of two new cells, or we often refer to them as daughter cells. So we get our DNA replication. And then it's followed by separation of the chromatids into daughter cells, which we know there'll be two. So from one cell we get two. Yes?
At what point would something have to be changed to have identical twins?
To have identical twins, typically when we actually are going into the blastocyst and morula stage, which we'll get into in a little bit, that's when we actually have the separation actually occurring. And suddenly we get two instead of one. Yeah. Let's go into, now we are fertilized, and now we are a zygote after fertilization, and we are in the pre-embryonic time period. That's the first 2 weeks after fertilization, and we have to make it from the ampulla all the way down the tube into the uterine body and implant in a nice appropriate area inside the uterine body. What's interesting is once you start mitosis, you keep undergoing mitosis. And so you have the two daughter cells undergo mitosis, and suddenly you go from 2 to 4. But this mitotic process that's occurring during this time of travel to the uterine body is not a typical mitotic process, and we call it cleavage because of that. And the reason it's called cleavage instead of plain mitosis is, as you remember, when a cell undergoes mitosis, we get two daughter cells and they grow, and then they undergo mitosis, correct? And then two more daughter cells and grow and undergo mitosis. Well, we are confined. And so what do you know about the size of the cells if you are in a confined space? Are they going to really have the ability to grow? And that's what cleavage is all about. We actually have a mitotic process without cellular growth, and all of the cells get progressively smaller instead of staying the size of the original cell. So cleavage is that rapid mitotic process following fertilization and results in smaller cells due to confinement by our ZP, our zona pellucida. So remember, it's a hard shell now. So we're going to undergo a lot of this mitosis, but the resulting daughter cells will be smaller and smaller and smaller. And so by 72 hours after fertilization, we will go from the original 1 fertilized cell to 16 cells. And it is so recognizable that it actually has a name. It's called the morula. And I want you to realize the morula looks like a berry. And like a blackberry or a raspberry or a boysenberry, it has all these little bitty cells all collected, all together, compacted. And that is known to actually happen by 72 hours. And so we are confined in this zona pellucida, but we all are about the same size, filling that area up, all much smaller than the original fertilized cell. Just drawing that. The interesting thing is, by day 5, so 3 days after fertilization, we're traveling through, and by day 5 after fertilization, we are now being released into uterine body, or cavity, the cavity of the uterine body, and floating freely, and ZP breaks down. Finally, it's worn down by all the trauma that it's also experienced going down the fallopian tube, breaks down and releases a blastocyst. So morula, berry-like, blastocyst, a hollow sphere with 2 cell types, a trophoblast, which forms the wall of the sphere, and an inner cell mass called the embryoblast, which is basically a cluster of cells along the inner side of one area of the sphere. Importantly, the trophoblast will become the placenta. And the inner cell mass, or the embryoblast, will become the embryo. Eventually. So day 3, we look like a berry. Day 5, we finally made it into the uterine cavity, and the zona pellucida has been actually roughened up by traveling down the fallopian tube, just like the sperm cells got roughened up by traveling upstream. We're still getting roughened up by peristalsis and ciliary action, and that's going to help to break down the zona pellucida, gets inside the uterine cavity, and by day 5, we are not a morula, but we are a blastocyst, but we're all crumpled up in the zona pellucida, and so once the zona pellucida cracks, we spring forth, as this blastocyst, a hollow sphere with an inner cell mass that's going to become the embryo. Now, in order to make it to the embryonic stage, we have to undergo successful implantation. You are not going to make it unless you successfully implant. And so timing is everything in this process. So the implantation of the blastocyst starts at day 6. We said day 5, the zona pellucida and its contents make it to the uterine cavity, break apart and release the blastocyst. Day 6, we begin our implantation. And we typically have successful completion of implantation within a week or less. Let's review our cartoon up here. Secondary oocyte, remember that means we're stuck in meiosis II if we're a secondary oocyte. In order to complete meiosis II, we actually have to have fertilization occur. That's why in this picture here we see 2 polar bodies. This one, polar body, is indicative of completion of meiosis I. So 2 polar bodies there means fertilization did occur. You see our pronuclei coming together a little bit more over here, and then the actual fusion, and then we have our zygote, the single cell, which doesn't stay a single cell long at all. It rapidly goes into its first mitotic phase, and we get our 2-cell stage. And it's trying to show you in this picture basically where this is actually occurring. So there's the ampulla for fertilization that's actually occurring. And moving on down the uterine tube towards the uterine body, it's showing you that the morula is still essentially in the tube getting ready to be released. Once it's released into the cavity and it springs forth from its zona pellucida, we have our blastocyst, and then the blastocyst is going to begin its implantation process. Where the inner cell mass or the embryoblast is located, on that side of the trophoblast are going to be some digestive enzymes that are going to be produced to allow it to actually land on the endometrium and start to digest its way to get buried within the endometrium. Now, this endometrial lining is called secretory. And the reason it's called secretory is because of a certain hormone. What built up the endometrial lining first after menses? What builds up the endometrial lining? That's stratum functionalis. Estrogen. So estrogen builds it up first, but what makes it lush or secretory? Progesterone. You remember in our diagram, progesterone peaks, doesn't it? So progesterone goes up for about a week, hits its peak, and if nothing happens, it starts to go down. And this process, this time frame, is really important because progesterone is important in creating the proper environment for implantation. And this pathway and this time frame for the fertilized egg or our zygote to make it to this area for implantation is very important, because if it gets there too soon, we don't have the proper buildup of the lining, and if it gets there too late, the lining is actually on the decline and it will be lost. Are you guys following me? This time frame after ovulation is very important, and so the fertilized egg must get into the endometrial lining at close to that peak for progesterone formation. And so these days are actually very specific for this implantation process. You get there too early, you don't have the proper buildup. You get there too late, you are actually losing your buildup and you actually are not going to be able to successfully implant. So let's look a little bit more at what happens when the blastocyst comes into contact with the endometrium. It's going to secrete these digestive enzymes that are going to allow it to burrow into the stratum functionalis. This implantation that's going to happen, this is called implantation, takes almost a week. Our notes here show day 6, blastocyst is going to come into contact with the stratum functionalis of the endometrium. Here's day 7, and now we see these digestive enzymes eating into the endometrium itself. And what's interesting is that, if I erase this, we take a look at our names of the trophoblast, and we still have, take a look here, we still have trophoblast and trophoblast with the names. But one is called cytotrophoblast and the other one is called syncytiotrophoblast. The digestive enzymes are eating away cell membranes. And in the syncytiotrophoblast, that is creating a luscious muck. This luscious muck is nutrients, because this journey has taken that really big, fat, nutritious egg and has pared it on down to a normal-sized cell that needs nutrients now. And that's another reason why this implantation must occur. It is now getting ready to starve if it doesn't get some nutrients. And so we have to burrow into the endometrium to do that. And our syncytiotrophoblast is actually like cytoplasm. We are digesting away the membranes of the cells, and it's like we have this cytoplasmic mush. And this is going to feed off of that. So this is a very important part of the implantation process. We have to preserve this embryoblast that's inside the trophoblast. So we get some cellular modification of the landing zone. And it's always where the embryoblast is, that's where I say the landing material is all there. So this area, it's not going to land on this side or this side or this side. It's going to land right here on that side. That's where all the landing material and digestive enzymes are going to be. Then as this process occurs, here is day 8 after fertilization. Here we are, and we have created additional now cell layers. And we are going to cause these cell layers to transform into additional structures, such as the yolk sac and the amniotic cavity. And those areas are going to be important in preserving and helping the development or the developmental process of the cells in between. This is what actually is going to now become the embryo. So the yolk sac is on one side and the amnion is on the other side, and in the middle is going to be the developing embryo, and that's present on day 9. As soon as we are starting to make this muck right here, the syncytiotrophoblast, HCG is being released. So day 7, after fertilization, HCG is being released. HCG is going to cause the corpus luteum to live for 3 months. As long as HCG is present, the corpus luteum will not degenerate into a corpus albicans. It will live as a corpus luteum, and it will produce progesterone. Because if you don't have progesterone being produced, you will not have maintenance of the uterine lining. Progesterone is absolutely important for successful maintenance of our developing embryo and then our fetus. We have to have maintenance of the uterine lining. Let's take a look at some of the other notes about implantation. It takes approximately a week. The blastocyst embeds itself, gets itself covered up by endometrium. And the syncytiotrophoblast is actually going to secrete HCG, which is going to maintain the corpus luteum, which is going to keep our progesterone levels high, which is going to maintain our endometrium. You know, I was trying to make a point. The number of days that this takes is exceedingly important, because if you get there too soon, you're in bad shape, and if you get there too late, you're in bad shape. And that's why we have this next story here. Most of our zygotes, most of our fertilized eggs, fail implantation. So 2/3 of fertilized eggs do not have an opportunity to actually become an embryo or to even make it to the embryonic stage. If they cannot implant successfully, they cannot survive. If they do not successfully implant, there is no HCG, and that means progesterone will drop, and that means menses, the next menstruation will occur. HCG is important for maintaining the corpus luteum. And the corpus luteum is important for progesterone, and progesterone is important for endometrial lining. So implantation is successful, and now we have the work to actually form the placenta as well as form the embryo. So our next step is placentation, the formation of the placenta. This can be considered an extension of implantation. Placentation, the formation of a placenta. It takes almost 3 months to actually form a mature functional placenta. The placenta is fetal life support, embryo and fetal life support. It is designed to be a temporary organ, and it connects our conceptus, whatever stage we are, to the uterus and causes amnesia. So causes immunologic amnesia. So amnesia is, you can't remember, don't know, no big deal. You know, if you don't remember, it's not a big deal. Immunologic amnesia. Whose immunologic amnesia? Moms or babies? Who's carrying the foreign material? Mom. So the placenta is going to be responsible for making sure mom's immune system doesn't know that there is a conceptus or a foreign body inside her. Because isn't it a foreign body? Absolutely. You know, even the zona pellucida was protecting this as it was traveling down the fallopian tube, wasn't it? Protecting it so that nothing from the immune system could actually get to it and to attack it. It's a foreign body, and the mother's immune system would actually attack it if it wasn't for how the placenta functions. Two important things about the placenta. Remember, it's temporary, which means after birth of the child, it's meant to be lost. And so we have two areas to pay attention to right now. We have the placenta has kind of a subdividing line between it. It has one called a decidua basalis and a chorion. And the decidua basalis is the maternal part. So in other words, this placenta is half mom, so to speak, and also almost half mom, and then half baby, because the chorion is the fetal part. What what's the decidua basalis going to be called in a normal uterine lining? Remember, the uterine lining has two, the endometrial lining has two components to it, stratum functionalis and stratum basalis. So what is the decidua basalis actually? Of those two. Is it going to be stratum basalis of the mother? No, stratum functionalis. So keep in mind when you see decidua basalis, that means that we have pregnancy occurring and mom's stratum functionalis is contributing to her part of the placenta. And so let's take a look at some of this stratum decidua basalis and the chorion that's actually going to occur. We're going to go down to this set of pictures right down here. It's trying to show you, here is our nice little embryo in the middle, and here is the placenta in its early stages of formation, and it's got these blood vessels coming off of it that are going to seek out mom's blood supply. So this is a parasite, actually. It's seeking out mom's blood supply. And this red stuff all around, these little, what are called chorionic villi, that's all mom's blood. All of this that you see on here indicating this, all of this on either side of this villus is mom's blood. And so remember I told you we have these enzymes digesting away. And that's the interesting thing. We will digest into mom's blood vessels, and she will not hemorrhage or bleed to death, because this area is actually designed for this, because this is stratum functionalis, which is actually designed to be shed, right? So it has a little bit of its own little safety net. So we're going to send off our little feelers, which is the chorion of the placenta from the fetal side, and we're going to stick our fingers in mom's gooey blood inside the decidua basalis, because mom's gooey blood is going to give us nutrients. It's going to give us oxygen, it's going to give us glucose, it's going to give us amino acids, it's going to give us all the stuff that we need to develop. And then we're going to be able to give her our wastes and our carbon dioxide and all of the things that we don't need that are basically waste materials. And so that's why you see in this little area here, we see red stuff and we see blue stuff. Because red stuff means mom's oxygenated blood, and the blue stuff means it is now becoming deoxygenated, which also probably means nutrient poor. And our arteries are feeding into this little blood pool, and because they've been munched through by the enzymes, but also the veins are feeding into this blood pool. But which is higher pressure, arteries or veins? Arteries. So we will act like a stream, even though we have an artery and a vein kind of close to one another, filling up this pool. The arteries will pump in the fresh blood, and because the pool is filling up, the used stuff will actually leave. So this little chorionic villus, which is vasculature that is going to take blood back to the fetus, is going to then bring back nutrient-rich, oxygen-rich blood. And that's going to be colored red, right? The good stuff is colored red, the fresh stuff. And the stuff that's not fresh is colored blue for us typically, correct? And so what you see in this chorionic villus is basically blood vessels from the placenta indicating that these chorionic villi are capillary beds that have fresh and used blood within them. The fresh blood is going to go where? Back to the fetus. The used blood is going to go out to the mom, to these little blood pools. Okay, that's what's really interesting about this. This mom's blood system, her blood is being exposed to this foreign material, but she contributes to this foreign object, the placenta. And this contributes to her immunologic amnesia about the whole process here. So these capillaries that are in these villi are slightly protected, but they're almost literally bathing in mom's blood, this capillary bed of these chorionic villi. The placenta takes the first trimester to mature, and in the beginning is going to be responsible in its early stages for HCG, because HCG is going to maintain the function of the corpus luteum. This is only going to be released for the first 3 months. After the placenta becomes mature, it no longer releases HCG. So what does that mean about the corpus luteum? It regresses, becomes corpus albicans, so progesterone from that feature is no longer going to be present. And that's because as the placenta is growing, it actually is also producing estrogen and progesterone. So estrogen and progesterone are going to be produced by the placenta throughout the time of its formation, and especially after maturation, and those hormone levels are going to increase throughout the pregnancy. And estrogen and progesterone that's going to be found is coming from, literally from the placenta, not from the ovary. And if estrogen and progesterone is coming from the placenta, what do you know about mom's FSH levels? They're down because we want them down, right? We're already pregnant. We don't need any more. And so estrogen and progesterone are also inhibiting FSH levels, keeping them very low, which means we're not having any follicles actually develop, and so no more pregnancies during that time. But the placenta produces estrogen and progesterone throughout its formation. It's just that those levels are low in the beginning, and then after maturation, those levels continue to rise throughout the pregnancy. And estrogen and progesterone are really important for typical normal growth and differentiation of both the mother and the fetus. Produced by placenta throughout the pregnancy. And increases as time goes by. Another hormone that's produced by the placenta early on and throughout the pregnancy is called HCT. HC, human chorionic, T, thyrotropin. What is the target organ for this? The thyroid gland. And what is it going to cause the thyroid gland to produce? Thyroid hormone. And what's going to happen to mom's metabolic rate? It goes up. And we have to have mom's metabolism actually increasing because she is actually caring for more than just herself. And we have to ramp up her metabolic rate to actually do that. I think we should stop there. And we'll do formation of the extraembryonic membranes and then embryonic development next time and finish off the pregnancy.